NDEWS. My name is Emily Lockhart, and I work for the U.S. Drug Enforcement Administration as a senior forensic chemist in the Emerging Trends Group. This video has been prepared in lieu of the annual presentations provided by my colleague, Jill Head, and will describe the data presented in our annual Emerging Threat Report. The Special Testing and Research Laboratory houses two programs that focus on new psychoactive substances. The Emerging Trends program performs analysis on domestic and foreign drug seizures and performs research and method development to aid in the identification and quantitation of NPS. We also publish quarterly emerging threat reports, which aim to monitor past and current trends. It's important to note that DEA laboratories perform analysis on seized drug material only. We do not conduct any toxicological testing on biological fluids. The Reference Materials Program works tirelessly to authenticate reference materials and authenticate SWIG drug monographs and the FTIR library offered by SWIG Drug. Both groups together work to perform structural elucidation of unknown drugs. For the rest of this video, I will focus on the data published in the 2019 Emerging Threat Report. This is a snapshot of page 1 of the Emerging Threat Report. As always, the Emerging Threat Reports will be available on the NDEWS website at www.ndews.org and are disseminated via an email distribution list maintained by the Emerging Trends program. The data for the Emerging Threat Reports are retrieved through a query of archived seizure and analysis information from drug evidence analyzed by the DEA laboratory system. It is representative of drug evidence seized and analyzed in calendar year 2019. Please keep in mind that this is a snapshot of the NPS market and is not comprehensive of all NPS, nor is it representative of all drug evidence analyzed by DEA. Prioritization of exhibits and laboratory backlogs impact the data presented in these reports. An identification is made when authenticated reference material is available for comparison. When reference material is not available, the drug evidence is identified as substance unconfirmed or inconclusive. There were 175 synthetic cannabinoid identifications in calendar year 2019. This represents a 200% decrease from 2018. 5-fluoro-MDMB pica and 5-fluoro-MDMB pinaca were the most reported synthetic cannabinoids, representing approximately 59% of the identifications. There were 16 different synthetic cannabinoids identified. 5 of these cannabinoids were reported for the first time in 2019. These include 4-fluoro-MDMB butanaca, 5-fluoro-AB pinaca, 5-fluoro-ADB pinaca, 5-fluoro-kumul pinaca, and APP butanaca. There were 184 cathinone identifications representing 19 different substances in calendar year 2019. This constitutes a 78% decrease from 2018. Eutalone was the most reported cathinone, accounting for approximately 36% of the identifications. 6 cathinones were reported for the first time in 2019. These include eutalone, BMDP, or 3,4-methylene dioxy N-benzylcathinone, N-butylpentalone, 4-chloro-N-N-dimethylcathinone, 4-chloropentadrone, and 4-fluoropentadrone. There were 5,381 synthetic opioid identifications in calendar year 2019. This signifies a 33% increase from 2018. Not surprisingly, fentanyl was the most reported substance, representing 81% of the synthetic opioid identifications. There were 27 different synthetic opioids identified. 6 of these opioids were reported for the first time in 2019. These include ortho and para fluorofuranylfentanyl, O-desmethylcis-tramadol, 1-benzyl-4-piperidine, which was identified in a foreign seizure, despropionyl orthofluorofentanyl, and N-benzyl parafluoronorfentanyl. There were 115 NPS benzodiazepine identifications in calendar year 2019, representing 5 different substances. Atizolam was the most reported benzodiazepine in this category, representing 53% of the identifications. There were no NPS benzodiazepines reported for the first time in 2019. There were 15 piperazine identifications in calendar year 2019, representing 4 different substances. None of these piperazines were identified for the first time in 2019. This slide summarizes the miscellaneous category of NPS. There were 94 exhibits where the laboratory was unable to conclusively identify the substance. This was largely due to the unavailability of reference material at the time of analysis. MMMP, or methyl-methylthiophenylmorpholino-propiophenone, was identified in 7 exhibits. The hallucinogens 2CB, 2CC, and 2CH were also identified in drug exhibits. One of the active components of kratom, mitragynine, was identified 4 times. 2 PCP-related substances, 2 oxo-PCE, and 3 methoxy-PCP were each identified 3 times. And 2 fluoro-deschloroketamine was identified 1 time. New for this annual report is the addition of graphs summarizing identifications of select substances from calendar year 2016 to calendar year 2019. In 2016, FUB-AMB was the most prevalent synthetic cannabinoid identified by DEA laboratories. 2017 and 2018 saw a mix of FUB-AMB, 5F-MDMB Pinaca, and ADB-Fubinaca. In 2019, 5F-MDMB Pica and 5F-MDMB Pinaca became the most prevalent cannabinoids. In 2016, dibutylone and N-ethylpentalone were the most identified cathinones. Throughout 2017 and 2018, N-ethylpentalone dominated the cathinone market. By 2019, dibutylone and ethylone had nearly disappeared, and utylone emerged as the most prevalent cathinone. The shift from one cannabinoid or cathinone to another is often triggered by a change in legislation in either our country or the source country. Fentanyl identifications consistently rose from 2016 to 2018. 2018 and 2019 saw a nearly identical number of fentanyl identifications. Acetylfentanyl identifications appear to be on the rise. This substance is often identified as a secondary component in fentanyl samples. 2 furanylphenol identifications peaked in 2017 and have seen a dramatic decrease. Likewise, 4 fluoroisobutyrylphenol identifications peaked in 2018 and saw a dramatic decrease in 2019. Carfentanil identifications also peaked in 2017, but have averaged about 50 identifications per year since that time. Atizolam has remained the most prevalent NPS benzodiazepine since 2017. We've also seen the emergence of other NPS benzodiazepines such as flubromazolam, flualprazolam, and clonazepam. In 2017, a class scheduling action was passed to control many of the fentanyl analogs. At that time, we saw a shift to identify new opioid structures that did not fall within that fentanyl class control. Subsequently, we saw an increased variety in NPS benzodiazepines on the illicit drug market. This graph represents synthetic opioid identifications pulled from the emerging threat reports over the last 5 years. It is a great visual to show the staggering increase in identifications, as well as the increasing variety of opioids seen on the illicit drug market. It has come to my attention that there are a lot of questions about what impact COVID-19 will have on local drug markets. At this time, it's hard to say what the impact of this virus will be. Forensic laboratories and enforcement operations have been altered in accordance with CDC recommendations, which will impact both the volume and timeliness of data describing the illicit drug market during this unprecedented time. Our laboratory will continue to monitor the trends and report them to you via the emerging threat reports. Thank you for taking the time to watch this video. I encourage you to direct any questions to the NDEWS Network, and myself or someone on the NDEWS team will respond.